The title of my presentation is Characterization of Plant Growth Promoting Rhizobacteria from Feed-Grown GM and Non-GM Maize. In the lab where I come from, our interest is in microbial biotechnology. We do fingerprinting. We work on plant growth promotion as well as biological control. And GM is also part of our interests, which include biosafety and biosecurity. Back to the topic. When we say plant growth promotion, we are relating to plants and microbe interaction in such a way that the microbe of interest are the ones that are going to enhance plant growth. Now, when we want to link it to the issue of GM, looking at plants the way it is, we know that plants excrete some things into the rhizosphere, which we call exudates. The exudate from the plants, it is believed that when a plant is a GM, probably the exudates may contain some things that may not be there in a plant that is not a GM plant. And that is what generated these studies. So part of the microorganisms that we have found before that are related to plant growth promotion are the ones that are listed here, which include Pseudomonas and many others, as shown in this slide. And the mechanism that these plant growth promoters use include the synthesis of phytohormones. They also make use of antagonism against pathogenic microbes, induction of host resistance, nitrogen fixation, as well as synthesis of antibiotics. For BT corn, for an example, it has the advantage that it can increase insect resistance. For that reason, grain will be enhanced and plant growth will improve. However, when you it is also possible that the BT endotoxin that is being introduced into the rhizosphere through the roots, as the exudate may influence microbial community at the roots. So we now want to look at the possibility that it influenced the microbial structure at the root, and that is the genesis. It was hypothesized that survival and functioning of microbial community associated with the rhizosphere of BT maize differs from its non-BT maize counterparts. And the aim of the study is to evaluate the effect of BT maize root exudates on rhizosphere community structure. We took a sample from Gauteng province in South Africa, and the samples were collected from BT maize and maize without the genetic modification. Samples were collected 30 days after sowing and also at post-harvest. We use this time duration because we want it to be that the plant would have excreted so much into the rhizosphere. By the way, rhizosphere is area around the roots. So 16 soil samples were collected in triplicates, and we did cereal dilution on the soil, plate the cereal dilution, incubate it, and after 2 to 3 days, colonies were selected. Now, the isolates were characterized biochemically using API kits, and we also test them for plant growth promoting traits, and they were also characterized molecularly using 16S primer and universal primer, as shown here, and our target region were 989 base pair and 1005 base pair region. I'm not going to bother you with this. Now, when we did the PGPR trait, we checked for indo-acetic acid production, production of ammonia, catalyst production. We did check for the antifungal activity phosphate solubilization, as well as hydrogen cyanide production. In conclusion, from what we observed, now when you are looking at this figure, try to compare the top one with the below one. Don't compare top to itself. It was found out that there was no difference between the GM and the non-GM in terms of the colony forming units that were observed on different agar media. And with regards to the API test that was done, we found out that the colonies that were common to the GM plants were also the colonies that were common to the non-GM plants. When the plant growth promoting trait was tested, it was established that hardly is there any difference. If a microorganism like Pseudomonas luteola, for an example, is able to show antifungal activity in GM plant from the GM rhizosphere, even when you find it in non-GM rhizosphere, so long as it is still the same organism, the same activity will be shown. And the pH value that were documented from what we sampled did not show any significant difference. We also did the biolog for microplates, and we found out that there were no significant difference in the optical density reading. This is also from the biolog, and there weren't any significant difference, be it at 30 days or at post-harvest for GM and non-GM. In this one also, you see the superscript that you see there. A is an indication that everything is A, whether it's a GM or no GM at 30 days or be it at post-harvest. We did not see any significant difference. And the region that were to be amplified when PCR was done for the GM rhizosphere and the non-GM rhizosphere, the region was still amplified in the polymerase chain reaction. This is similar to the previous one I've shown, but the area of amplification were different. We did the blast search just to see the percentage similarity that we have from our work and what we have in the gene bank. And the result is as shown here. Common to the rhizosphere in the GM plant are bacillus. We also found bacillus common to the one in the non-GM rhizosphere. Now, I've tried to wrap it up briefly, but in final conclusion, what we found out is that most rhizosphere isolates from the GM and non-GM maize rhizosphere, both of them showed plant growth promoting traits, which is our interest. Then, looking at it at the molecular level, genetic modification of maize, so far that we have seen, had no effect on bacterial community in the rhizosphere. I would like to say I acknowledge members of my research group and also Dr. Barros for giving us assisting us to get the samples, and also National Research Foundation. Thank you for listening.